Good morning, everybody. Welcome to today's biological physics, physical biology seminar. So today we are very excited to hear from Camelia Prodan and Ala Trusina about their work on biological physics. Without further ado, Camelia, you're the first speaker. Please take it away.
Yes, thank you so much for inviting me to give a talk here. It's an extraordinary seminar series. I'm very happy to participate and to attend. So today I'm going to talk about topological mechanics and possibilities of applying what we learn from topological mechanics in biological systems. My main talk, the main purpose for this talk will be to inspire you, the researchers, to look for topological effects in biological systems in order to possibly discover new biology. The work I'm going to present today is done by a very talented group of people, students and postdocs, and some of them are in this picture here, holding of what we call a microtubule. Okay, so the overview for my talk will be, I'm going to talk about the microtubules from the point of view of topological mechanics. And I may make the case for, or not, for searching for topological signatures in biological systems. I'm going to introduce later what topological modes are, and especially topological edge modes, and what lesson from topological metamaterials we learn and possibilities of applying them in biological system. And I am going to end with some experiments we've done also on microtubules. So I'm going to start presenting the microtubules from the biological point of view. The microtubules are really engineering marvels. They are very thin, only 25 nanometer in width, and very stiff compared to their width. They are stable in aqueous environments. They assemble and disassemble, and very importantly, they maintain the cell shape while dynamics, and they are very essential to many cellular processes and morphologies. So this is a picture of a cell where the microtubules web are labeled in red. They extend from the nucleus, here labeled in blue, all the way to the end of the membrane. If we zoom in of one of these microtubules, we will see that they are actually polymers made out of the protein called tubulin, which is in a dimer, form. Well, how do we make these proteins, these microtubules? Well, we have the tubulin dimer, right here, the alpha and beta tubulin. We put it in a special buffer, and the proteins themselves are going to self-assemble into a protofilament. The protofilament self-assembles into a sheet, and the sheet then closes into a tube, and this tube starts growing, and at a totally random time, the tube will start falling apart until a certain point, and then it's going to start growing again. The main point of my discussion is to actually argue that it's possible the topological edge modes are the ones that are responsible for the microtubules falling apart. So before I start talking about topological modes and what they are, I'm going to explain what actually can we do with topological modes. What are they good for? From this picture, this is an experiment that we've done on a tabletop setup. And these are regular normal modes. These topological modes are huge. They have very large amplitudes, meaning they can store a lot of energy. They are very efficient at storing energy. And again, thinking back to the microtubules, you can think of this energy, the energy coming from the GTP hydrolysis. Topological edge modes are only existing at the edge or at the seam or at interface. This is a profile of a topological edge modes where only the end is activated, while in a normal mode, every single particle in the system is activated. So the energy that comes from external sources remains at the edge, while in the case of a normal mode, the energy just gets distributed absolutely everywhere into the system. Topological edge modes are very efficient at energy transfers. You can transfer the energy from one point to the other point of the system by doing some very simple shifting. And this energy is not going to go into the whole bulk, but it's just going to go along the edge of the system. And maybe an important part, it's that these topological materials are very easily tuned. You can have a topological mode, or just by a simple small stretching of the material, you can make those topological edge modes disappear. You stretch them back on the original form, and the topological modes appear again. Now, topological mechanics in general didn't exist 10, 15 years ago. It's a new field of studies that is very energetic. A lot of people are interested into it. And it's inspired from the highly studied topological insulators, which are quantum systems. Briefly about topological insulators, usually if physicists want to devise a new material that have these amazing properties, they have to go to extreme conditions like very high magnetic field or very cold temperature like superconductors. In the case of topological insulators, these very high magnetic fields are somehow built in the lattice of the material. So you don't need to apply external stimulus to obtain this type of topological insulators. What is very interesting, and if you talk to physicists 15 years ago, they will not believe it. You can take this quantum system, respect the symmetries of the system of the lattice, make them in classical system. And initially it was very difficult. People didn't know how to make them, but now you can make them out of Legos. The topological modes are so robust. They appear, as long as you respect the symmetries, they appear in any type of system, acoustic, elastic, magnetic, Legos. So in this case, in classical system, instead of electrons, we talk about phonons. And now many of the lattices that are discovered for topological insulators already have analogous into classical systems, like the quantum Ballet Hall effect, for example. You can make it on a tabletop in your living room right now and study it. Again, these are not quantum properties. The properties are similar to what you observe in quantum, but they are classical properties. Now, topological mechanics has really revolutionized the material science, providing new materials that are targeted at steering energy and storing energy. They are very efficient way of storing energy. And depending on the patterning, the topological mechanics in our case, in our studies, only depends on patterning. We have used the same pattern into acoustic system and magnetic systems and Lego systems. You will see exactly the same effect. And we propose our own idea is that it's possible that nature actually stumbled on those patterns. You'll see the patterns are not crazy. And they could use actually these effects of topological edge modes to do biological work. Now, studying topological effects in biological materials is not easy. It was not easy even in tabletop systems because we didn't know how those modes look, what can we do with those modes. And studies with every single tabletop experiment we've done, in my case, only in my case, was done with the microtubules in mind. learning what could the microtubule do in this situation. All our work is on resonators. Anything that resonates, we can use it. One of the very familiar one is a fidget spinner that we have. Another one, we have acoustic systems, which are really tubes in which if you put a speaker, you can generate modes. If you couple 3 of them, for example, this mode, the first mode will split into 3 modes and so on. But I would like to make the case also that proteins, which are classical systems, are also resonators. If you put proteins together, they also interact, they couple together through either hydrophobic or van der Waals interaction, resulting in resonators. So every time I am showing you movies with the fidget spinners, please keep in mind microtubules or tubulins are also classical systems. They obey the same laws of physics. And you could imagine replacing them for the purpose of understanding what can we do with these topological edge modes. Now let me start from the beginning. This is just an intro to normal modes. If you have a string, if you have a string and you shake it with a given frequency, at one frequency, you can generate the first mode. You increase the frequency, you generate the second mode, the third mode, fourth mode, and so on. These are called normal modes. And what's special about them is that all the particles in the system, that frequency, they move with maximum amplitude. Yeah? Whether it's a lower frequency of the first mode or the slightly higher frequency of the second mode, all the particles in the system will move with maximum amplitude. We can look at a system of points, a discrete system. These are just points connected, for example, by a spring. Every time we want to talk about a spring, we actually really talk about harmonic interaction and harmonic potential. And in a system of a ball and spring, for example, you can generate also modes where the ball will move according to the first, second, third mode, and so on. And again, the modes here, the resonant modes, the motion of the amplitude of the motion is maximum for every single particle in the system. If you take these modes and you plot them as frequency on the x-axis, you obtain what is called, you obtain what is called the Phonon Spectrum. So this is just a collection of the normal modes. Now, this is just a drawing of a Phonon spectrum. They have different shapes. And some materials have a gap in between their normal modes bands. And this gap is actually prohibiting the propagation of any type of energy within the system. So if you take a material like this one right here, if you shake it with this frequency, nothing will happen, absolutely nothing. Many materials don't have this one. So if you just take, for example, a system of balls with an equal distance in between them, their gap will disappear. In here, you can excite them with any frequency from their Phonon spectrum. The moment you dimerize, and this is very important, the moment you dimerize a system, and dimers are everywhere in biology, you open this gap right in here. Now, what topological mechanics did a few years ago, they say that in this gap, some materials have one resonant mode, and this resonant mode is called topological edge modes, and it only exists at the edge of the microtubule, of the edge of the system, and it will not propagate inside the material. Now, let me show you some examples of topological edge modes. Again, we are using fidget spinners, toys like here. They are more sophisticated, but similar to what you have at home if you have kids or cats. And I would like to show you how a resonant mode is. So in both movies in here, we have a dimer. There are 2 degrees of freedom in this case, and you should have 2 resonances. I'm exciting the system here, just increasing the frequency over time. And you see, as I increase the frequency, the system moves a little bit, not too much. But when it hits the resonant frequency, the system moves a lot. You continue increasing the frequency and the system starts to calm down again. So only at that resonant frequency, not below, not above, the motion of the spinner is very high. These here are out of phase resonances, meaning the spinners are moving towards each other, while this one is called the in-phase resonances, where the spinners are moving together, left and right, the acoustic and optic bands for the system. Now, this is the most scientifically described fidget spinners. We actually have a ball bearing and we have arms. And depending on how many arms you want, we can modify the moment of inertia for the spinner. And on these arms, we attach magnets and we can couple this in different systems. This is actually a video of what a bulk mode, a gap mode, and an edge mode looks like. This is a real system. Because the frequency is higher, we slow-mo the movie 4 times. So the spinners are actually moving 4 times bigger. You can go on Amazon, buy fidget spinners and buy magnets, and you can realize this system in your own home. You don't need anything special. Detail for this paper, these are actually Majorana modes that we expect are in this manuscript right here. So these spinners are attached to a table. They are only allowed to rotate, and they are connected to each other through magnets. And what's different in here is the length of the interaction. Some magnets are closer together. Some magnets are far apart together, depending on the pattern we want to realize. This is called the top one, for example, it's called the SSH, the Sush-Schrieffer model, where you have dimers, 2 of them together, connected by a weaker connection. And these dimers are connected to each other by stronger connection in here. And you put 2 of these SSH models together and you form a ladder. So you actuate only one mode right here. That's it, one spinner right here. You have an actuator, and the energy for the actuator from a bulk mode, a mode from here, it actually gets distributed everywhere into the system. Every single, pretty much fidget spinner except the nodes are moving. This one will be, for example, a node that's not moving. If you excite it with the frequency from the gap right here, nothing moves. We are exciting it here with that frequency, nothing moves. If we hit the frequency of the edge mode, of the topological edge mode, the energy from the actuator only stays here at the end. The bulk modes, the edge modes are actually very high, so we had to lower the amplitude so we can record it. So this energy only stays here at the end of the system, and it does not penetrate into the system. And if you let the system resonate freely, that energy is going to increase and increase and increase. This is a 2D system. This is what in quantum mechanics is called a Ballet Hall effect. It's really made with fidget spinners. And the pattern here is a pattern of graphene, which is dimerized, meaning there are fidget spinners that have 6 arms and fidget spinners that have 3 arms, put it in a honeycomb lattice. And this is a resonant mode, a bulk mode. We actuate from here just with a magnet that you could buy from Home Depot. And you can see the whole system. Again, this is not a simulation. This is a movie of what actually happens in the lab. This system is about 4 feet in length. And most of the fidget spinners, except the one of the nodes, are actuated. You try to actuate the one at the in the gap area, and most of the fidget spinners do not move. The reason I wanted to present this one is because you can generate an interface. Now talking about interface and going back to the microtubules, what I'm thinking of, it's a seam of the microtubule. It's an interface. There are similar stuff left and right, but there is a slight mismatch at the interface. The way we generate interfaces in this system, we just remove the arms of the fidget spinners. So if we want the energy to propagate this way, all the fidget spinners here have 3 arms. Of course, this one comes from mathematics. We don't do it randomly, but the movies are very nice and telling. So we can actually steer energy where we want in the system without the energy penetrating into the bulk. You can make zigzags in here, and you'll see that actually the vibrations will take corners from, will follow your pattern. Now, the topological edge modes, another important thing about them is that the topological materials are like magnets. You cut a material, you cut a magnet in half, you get 2 magnets, right? You don't get a north and a south separately, you get 2 magnets. The same with the systems of topological resonators. You change the interface, you make the system bigger or smaller, the new edge will get the properties of the old edge. And this is a system. You have a small system, you see the edge modes, the topological edge modes here on the edge. You only actuate right here. And these are the 2 edge modes. This is how the system is designed. If you increase the size of the system, you will have, this will be the old edge, nothing happens there, but the new edge has the properties of the old edge. If you increase it even more, can you see which fidget spinners are moving? Just these 2. And you only actuate right in here. So you can transport energy without losing too much to the bulk. Imagine if you had to transport energy from this corner to that corner. If you shake the whole material, there is going to be a lot of loss in here. But with this type of topological insulators, topological materials, you can actually transport it much more efficiently. And this is a ring-shaped system, similar to the microtubules, where the topological edge mode sits here at the seam, at the mismatch interface of the tube.
Camelia, you have 3 minutes left.
Yes, thank you. What can we do with the Phonon spectrum? Now, we can actually, by measuring the vibrational modes of the Phonon spectrum, we can measure the interaction between every single system, particle in the system. And if we go to the microtubules, for example, this is a paper we published in 2009. You can look at the lattice of the microtubule and map the interaction between the tubulin dimers as elastic constants, and you can get this Phonon spectrum. And theoretically, it's shown that this Phonon spectrum actually supports edge modes. Now, my last point of the system is that I like to introduce a phonon microscopy, the hunt for topological effects, but in general for normal modes into biology, we already measure bending modes, so we can get persistence length and Young modulus, but you can use these modes also to map the phonon spectrum of the microtubule. This is an experimental map of the phonon spectrum of the microtubules that show that actually the microtubule is not an elastic tube, but a pre-stressed elastic tube, which we knew obviously from before. Now, how can we measure this topological, this phonon spectrum? You can use, again, you can learn from colloidal works, and you can track the position of the microtubules as a function of time. You do some math that's described in this paper right here, and you can get the density of states or phonon spectrum for the microtubule. In here, the actuator is actually the water, but we can actually use electrical actuators and sensor because there is so much loss of energy into the water, you have to measure very closely to where you actuate it. So we are working in actually trapping a microtubule in a nanochannel and using electrodes to stimulate, to actuate the microtubules to measure the phonon spectrum. Well, it is a very difficult part. We don't actually need the phonon spectrum. We need to know where the gap is. So anyway, any other way of measuring phonon spectrum will be very useful, or any other way of measuring the topological edge mode or the frequency at which the topological edge mode is will be also very useful. Thank you so much.
Thank you, Camelia, for a wonderful talk. I see there are some questions, so I'll ask you the questions. So the first one is from Nancy Ford, and Nancy asks, she says, this fidget spinner lattice model looks really cool. Have you considered using this type of model in an undergraduate lab course?
Oh yes, we also use it for elementary school children. When we assemble very large lattices, we get them in the lab, and they actually assemble the lattice for us. But yes, in an undergraduate and graduate level, and yeah, it's very useful in learning Lagrangians and Hamiltonians and what we can do with them.
Awesome. And then Jinwa has a question, and I have a related question. So I'll first ask Jinwa's question, and then I'll state mine. So Jinwa says, quite a few years ago, I and Eva Nogales published a few papers showing microtubules have mechanical instability due to two types of lateral interactions between tubules. Here, I think you talk about smaller scale vibrations. Do you suggest these modes have functional implications? Can you elaborate on it? And
yes.
Yeah.
So I would say that I do not have any proof besides the theoretical work. I don't have experimental work, but I believe that the reason I want to do these experiments is microtubules can be triggered by this topological edge modes. The microtubules also can transfer much more efficiently energy within the lattice of the microtubule if it needs to. And probably this work can go even beyond the microtubules to enzymes that do work, and they have to get rid of their heat, which are phonons, in a very good way. So I do believe, and that's the purpose of my research, to show that this topological edge mode could have functional implication. And the idea we have for the microtubules is that the topological edge modes are the one that triggers dynamic instability by storing GTP hydrolysis energy in the end. When enough energy is stored, the microtubule will just explode from the edge. Just imagine huge vibrations that increases with the GTP hydrolysis, and at some point they are going to explode.
Yeah, and I guess so my question was kind of related again a little bit, like the interplay between the kind of, modes you're seeing, topological modes you're seeing, and the possibility of interplay between these and, dynamics of microtubules like dynamic instability and other things. And I don't know, I mean, do you have any comment or like, is it something you think might, like be a trigger or something?
Yeah, so we believe that these topological edge modes, whether on the edge or along the seam of the microtubules, are possible candidates to trigger dynamic instability. Once you figure out the frequency at which they act, technically you should be able to trigger dynamic stability or depolymerization whenever you apply that frequency. And again, as I showed previously, these lattices can be tuned. Right. If the microtubules or the cell doesn't want to depolymerize, it can attach these microtubule-associated proteins or other chemicals to either stretch the lattice or to change the configuration of the lattice. When the microtubule has to depolymerize again, those can come off, and you can start again.
Thank you, Camelia, for a wonderful talk and for these wonderful answers. So we will take more questions for you at the end of the second talk, once both talks are over. So thank you again, and we will move on to our